Good afternoon, children, and welcome back to biology class. Today we'll be saying about the blood cells, blood groups. Okay, right. Now, what is blood made up of? It is made up of plasma and formed elements. Right. Last class we saw about the plasma. Okay, now today we'll be seeing about the formed elements, which are the blood cells. And what are they? The RBC, erythrocytes, WBC, the leukocytes, and the platelets, which is thrombocytes. Okay, now coming to RBC, the erythrocytes. Now RBC is red in color because of the presence of hemoglobin, and the normal level of hemoglobin in an adult, in an average adult, is around 12 to 16 grams per 100 ml. 12 to 16 grams per 100 ml is the normal hemoglobin count. Okay, and the lifespan of the RBC is around 120 days because it has no nucleus, right? Its lifespan is only 120 days. And the normal number of WBC in an adult human being is around, in a male, is 5 to 5.5 million per millimeter square, cubic millimeter, sorry. So how much is that? 5 to 5.5 million per cubic millimeter of blood. And in females it is 4 to 4.5 per millions per cubic millimeter. Okay, and you know the blood is formed from, the blood itself is formed from bone marrow. You know what is the bone marrow? Inside the long bones there is a liquid like thing which is called the bone marrow, and it is the one which produces the blood by a process called as erythropoiesis. Okay, right. Now what is the shape and size of this RBC? You know, it is biconcave disc shaped without nucleus and mitochondria and Golgi complex, etc. It's all absent in RBC. Okay, and you know the function of RBC: to transport gases, mainly CO2 and oxygen. Now coming to WBC, the leukocytes. Okay, they are colorless because there is no hemoglobin, or it is colorless, you can say. And its lifespan is about 1 to 15 days, and every time new WBCs are getting formed. And the normal count of WBC in a human being or adult is around 6,000 to 8,000 cubic millimeter of blood. 6,000 to 8,000 per cubic millimeter of blood. Okay, they are also formed in the bone marrow and also in the lymph glands and the spleen. Okay, now structure is, they are nucleated and there are different types. We discussed last class what are the different types of WBCs. Okay, right. And what is the function of WBC? The main function is to provide immunity to our body. Now coming to the types of WBCs, these are the percentage of these different types of WBCs in our body, and we will be saying the functions also. Right, they are, you know what are the WBCs types of? Yes, neutrophils, acidophils, basophils, lymphocytes, and monocytes. So neutrophils are phagocytosic in nature. All these 5 types will help in providing immunity, but each of these have the separate functions to do. So neutrophils are phagocytic. Phagocytic means phagocytes means big eaters, right? And it will help to eat the germs or the antigens which are entering the body. Phagocytosis. Okay, and eosinophils help in allergic reactions. Children, those who have asthma and wheezing and allergic problems and all, their eosinophil counts will, or percentage will vary definitely. Okay, so resist infection, causes allergic reactions. You know, allergic reactions: sneezing, readiness of the nose and face, all those itching, all those are the allergic reactions. Okay. Basophils, they secrete histamines, serotonin, heparin, and causes inflammatory reactions. When a mosquito bites or when an insect goes through your skin, you find reddish rashes, right, and scratching on those areas. This is due to the reaction of release of histamines and serotonin from the skin. Okay, right. And monocytes, the neutrophils and the monocytes are phagocytosis in nature. They are the big eaters. Phagocytes means big eaters. They help in engulfing the germs which are entering the body. Okay, and the lymphocytes, as you know, they are B lymphocytes and T lymphocytes. They form the main immune responses of the body and produce the antibodies. These are the ones which produce antibodies, which will actually bind to the antigens. Okay, right. Now coming to the platelets or thrombocytes, they are also colorless. Their lifespan is about 7 days, and their function is thrombocytes. They help in blood clotting. Okay, and they are formed by megakaryocytes in bone marrow. They are formed again from bone marrow, of course, but they are formed from special cells called as megakaryocytes. Okay, and they are non-nucleated cell fragments. They are also non-nucleated, no nucleus, right? And we'll see blood coagulation. Now, how does blood coagulate? You can see when an injury happens on your body, a small cut or bruise or an injury, you can see the blood comes out. Blood starts oozing out. And within a few minutes or few seconds, the blood stops flowing out, and you can see there is a clot-like thing formed near the injured site, which is called as the actual clot. Now, how is this clotting or clot formed? We'll see that. Children, at the site of the injury, there is a clumped up platelets and tissues. Okay, and they release an enzyme called as thromboplastin. Okay, and many other factors are released. There are many factors present in the blood which are released at the site of this injured site, that is at the site of injury. And this is the simplest kind of reaction, where finally leading to clot formation. Okay, now the broken or the clumped up platelets will release thromboplastin, which will be converted to prothrombin. Thromboplastin to prothrombin, which is helped by the enzyme called as thrombokinase. Okay, thromboplastin gives to prothrombin by thrombokinase. Okay, and prothrombin again gives rise to becomes thrombin. Same enzyme is used here. Thrombokinase is used here. Prothrombin to thrombin, which will convert fibrinogen to fibrin. This is the simplest reaction, children. I said in short, prothrombin to thrombin, and this thrombin will help in converting fibrinogen to fibrin. And this fibrin is the one which is formed as a clot. You can see here, isn't it? A clot-like thing is fibrous. There are fiber-like structures in which the blood cells are getting caught. It cannot escape out. And you can see after the blood has got clotted, you can see very clear liquid coming out of your wound or the injury. Have you seen it? Yeah, that is the blood cells are getting caught in the fibrinogens or the clot, and the liquid, the plasma, is coming out of from the clot site. Okay, so fibrinogen to fibrin, which will finally form the actual clot. Okay, right. Next we'll see the blood grouping. Now there are many kinds of grouping done by many persons, and the very famous one most accepted by us is the ABO blood grouping. Okay, ABO blood grouping means what are the different groups, blood groups present in human population, children, which is the A group, B group, AB and the O. Okay, and also there is one more grouping which is the RH positive, RH negative. Okay, this blood grouping was found by or were discovered by Landsteiner or Carl Landsteiner. Okay, now how this grouping is done? On what basis is it done? It is done based on the presence of antigens. Presence of antigens which are present on the RBC, surface of the RBC cells. Suppose you can see the diagram itself. In this case, they have represented one type of antigen as blue dots. Okay, this is one type of antigens, right? But if that type of antigen is present in the RBC surface, they are called as A group. Okay, see here, this is another type of antigens. Okay, antigens are nothing but polysaccharides or protein bodies present on the RBC surface. Okay, now this group is called as, this is different from the first one. Okay, so this is now called as B group. Okay, now see the third one. Third one has got both the types of antigen. It has got. See, like the first one and the second one, both the A and B type of antigen is present on the surface. Now, what will call this blood group as? Of course, it is the AB blood group. And see the last one, on the surface of RBC there is no antigen at all. So how will you name what group these people will belong to? They will belong to a group called O group. O group means zero group, nothing actually is O group. Okay, right. And children, their respective plasma contains A blood group persons will have A antigen on the surface, and they will have in their plasma antibody against B. Anti-B antibodies they have. Same way, what about B group? B have antigen B on the surface. So what will they have in the plasma? They have anti-A. Okay, right. Now coming to the third type. What will these AB group have? If they have anti-A, and that will clot this, they have antigen A on the surface, right? That will clot the antigen A. If they have anti-B, they will clot that antibody will clot the antigen B on the surface. So these blood group people do not have any antibody against these antigens. If they have, it will itself get clotted and the person will die definitely. Okay, and what about this last group, O group? O group has no antigens on its surface. So they have both anti-A and anti-B in its plasma. Is it clear, children? Blood grouping. Okay. This column gives you which all group can donate blood to whom all, and who all can get blood from which all types of groups of blood. Okay. Now why this O group blood is called as universal donors? O group persons are called universal donors as they can donate to person with any blood group because they don't have antigen. That's the reason. They don't have antigens. They can give to anyone. Okay, so the person who has been given the blood, their antibodies will come and attack this blood. But here there is no antigen, right, children? Got me? So why they are called as universal donor? Because they don't have any antigens on its surface. And which group is called as universal acceptor? The AB group. Okay, and because they have both the anti-A and anti-B. Okay,